the Kirby Bauer disc diffusion method to test antibiotic sensitivity of different microbes. So in this particular case, you isolated microbes from your body in a previous lab, and then we would take one of those microbes from your skin collection and expose it to different antibiotics using this test to see if the microbe has sensitivity to a range of antibiotics. The first procedure in this workflow is to swab your microbe from a suspension, from a broth or liquid suspension, onto your plate. Usually we use a plate like TSA because a wide variety of microbes will grow on it. Some labs like to use, or doctors' offices like to use blood agar, and some will use a standard medium called Mueller-Hinton agar. In our particular lab, we use TSA. The goal here is just to give the microbes a nutrient-rich environment that they can grow on. So using aseptic technique, you get a swab full of microbes. The microbes are swabbed onto the plate. And the key here is to get a nice even suspension of microbes on the plate. So that's why the plate has to be rotated a few times, just to make sure that there are no gaps. You want to paint the microbes on all over the surface area of the plate, because again, we want a nice evenly distributed lawn of bacteria. We've labeled the plate in quadrants. Each quadrant represents where an antibiotic disc will be laid. So we use some sterile tweezers and testing your chopsticks using skills. Pick up a disc, tap it down, and each of those discs has a small amount of antibiotic impregnated into the paper. And each time flame sterilizing the forceps just to prevent any kind of cross-contamination. So now you can see how the samples are inoculated. I'm sorry, the discs are added. The plates are both inoculated and labeled. There's a control well up here just to let the microbes grow, really just to see how well they grow in the absence of antibiotic. And then these plates can be incubated. Usually it's for 24 hours. In this particular case, the individual is plucking the plates out of the incubator and doing sort of a time lapse of the microbes growing on the plate, so you can see these zones of inhibition that form around the discs. Oops. And so again, these zones are what you would measure and compare to a standard table of measurements that will then tell you whether the microbe is sensitive or resistant, or some intermediate value in between that. And then based on these zone diameters and your comparison to a table, you can determine whether or not, for example, this would be a suitable drug to prescribe if an individual is sick with this particular microbe.